Okay, so we have taken this sugar molecule. Um, we have broken it down. We have split it up into two molecules of pyruvate. We got a little ATP out of that. We then broke this down to carbon dioxide. We got a little ATP out of that, but we got a bunch of reduced electron carriers. So we have 10 NADH and two FADH2s. Um, and now finally we get to uh, pull out the energy. So this is lots of energy, but we have to figure out how to get it. And so the electron transport chain allows us to pull out the energy of those electrons in order to make ATP, and it utilizes the mitochondria. I went over this in the last lecture, but the mitochondria is a dual membrane system. It has a permeable outer membrane and a semi-permeable inner membrane with all of these kind of projections in it. And what we will see is that, and so within, let me see if I can get a lighter blue here. I don't know. I just feel like drawing today, I guess. Come on. So within this is the matrix, and then on the outside, I guess I'll do this pink here, is what is called the intermembrane space of the mitochondria over there. All right. This is uh, this is why I don't draw. So here's a look at that, and then the electron transport chain is on the surface of the inner membrane, and it actually spans the inner membrane of the mitochondria. And so because this is semi-permeable, it allows for, it blocks the flow of protons and other ions or charged molecules between the matrix and the inner membrane. Let me make that brighter. The matrix and the inner membrane of the mitochondria. I will say, you guys are all young, so this doesn't apply to you yet, but it's coming. One of the things that happens as you get older is that there is increased permeability of this inner membrane, which makes this whole system less efficient. And so you don't get as much bang for your caloric buck. And so this is, oh, whoops. Um, this is the a look at the electron transport chain. And again, just like photosynthesis, or at least similar to photosynthesis, what we're doing is taking high energy electrons. In this case, those high energy electrons come from NADH and not energized from the energy of the sunlight. And they're using the energy of those electrons to pump protons from an area of low concentration to an area of high concentration. Now, you remember from your talk with Dr. Handy that this is what's called active transport, right? When you have something that's going against a concentration gradient, that's active transport. And so we need the energy of electrons to push those protons out of the matrix into the intermembrane space. That proton gradient is a potential energy. And if you remember from the enzymes lecture, it's similar to a hydroelectric dam, uh, so that there's a buildup of that energy across that intermembrane space. Across that intermembrane. We actually call that a membrane potential. And the only way that those protons can get back into the matrix where they want to go is via the enzyme ATP synthase, and there it uses the energy of the downward flow of those protons in order to make ATP from ADP in inorganic phosphate. The other thing, and this is a big one, is that when this electron is done and has used up its energy, something has to happen. Now, in photosynthesis, it gets re-energized by sunlight and goes into NADPH. Here, this low energy electron gets picked up by oxygen to form water. Now, note this is the first, go down here, first and only time we use oxygen, right? The role of oxygen is to pick up this electron after it has given its service and its duty to drive proton uh, transport across the inner membrane. So it picks up that low energy electron and forms water in the process. So the oxygen that we breathe in is used to pick up these electrons and form water. Those protons are potential energy. I just said that. This process we call oxidative phosphorylation. Phosphorylation is the addition of the phosphate group to something, and oxidative is doing this in a way in which we, one, use oxygen, obviously, but we're using electron derived gradients. So this is different from substrate level phosphorylation. Remember, substrate level phosphorylation is when an enzyme directly pushes a phosphate onto a compound. So, two parts of this. One is the electron transport chain that we just went over, and then there is the actual use of the chemical gradient, the proton gradient here. We call that chemoosmosis, and we'll see why in a second. So, there's a lot going on here. Remember, this one is like 50 kilocalories per mole or something like that. I should probably look and see, go back. But lots of energy, right? We're breaking this up quite a bit, and the reason is because if we captured all that energy at once, then we would blow up the cell. Like we don't have a mechanism to pick up all that energy at once. It's kind of like a nuclear reaction or something like that. So we have to break it down and kind of sip that energy slowly. So the electron transport chain, as you said before, it occurs on the surface of it's embedded in the inner mitochondrial membrane. It has four protein complexes, three of which are transmembrane, so it spans the membrane. One kind of sits on the membrane. It also uses a couple of electron carriers, cytochrome C and ubiquinone. Um, And this should be animated. It's not, but that's okay. This is the process here. This is complex 1. This is complex 2, complex 3, and complex 4. And if you look at the flow of electrons, it occurs like this. NADH passes its electrons to complex 1. In the process, NADH gets oxidized to NAD plus, and it's available to pick up another electron via either the citric acid cycle or pyruvate oxidation. That electron flows through complex 1 to ubiquinone, which we abbreviate to be Q. At the same time, FADH2, that's produced in the citric acid cycle, passes its electrons on to complex 2 only. And actually, we'll see this in biochemistry. This complex is succinate dehydrogenase, and it is the same succinate dehydrogenase that's from the Krebs cycle. So this is actually directly passing its electrons from FADH2, and that's generated in the Krebs cycle into the electron transport chain. But that's for later on. Um, it also passes it on to ubiquinone, not at the same time. So this is a different molecule of ubiquinone, but both NADH and FADH2, those electrons end up in ubiquinone. Ubiquinone floats through the membrane and passes the electrons on to complex 3. From complex 3, these electrons get passed one at a time to cytochrome C. Cytochrome C sits at the surface and just kind of floats back and forth between complex 3 and complex 4, passing those electrons one at a time to complex 4, where ultimately these electrons end up linking up with oxygen to form water. Now, we'll see this a little bit later, but Um, in each of these transmembrane proteins, complex 1, complex 3, and complex 4, you will get the transport of protons through this from the matrix into the intermembrane space. Complex 2 doesn't transport protons because you see it's not transmembrane. And so there's a flow of protons in complex 1, 3, and 4 that allow for protons to leave the matrix and go into the intermembrane space. The result of that is to form a lot of protons here and to have a relatively low concentration of protons there. And so this is again a delta G kind of plot here of the energy of these electrons. And as I said earlier, as these electrons flow through the electron transport chain, it loses energy. So this electron in complex 4 is of lower energy than this one in complex 2 or coming in complex 3. And that energy is being used to drive those protons. This low energy electron here must be picked up by oxygen um, because electrons are powerful stuff and they just can't sit around like that. And so that electron is used to reduce oxygen to form water with a couple of other protons that is floating around in the matrix. So we call these guys pumps because they are pumping protons from an area of low concentration to high concentration, right? And you need energy in order to do that. So just like a pump in your basement or something like that, it takes energy, electrical energy, and pushes water against the flow of gravity. And this is in the look of an actual microgram of the matrix of the inner membrane of the mitochondria here. Pretty neat. And then now that we've created this proton gradient here, high proton concentration here, low proton concentration here, the protons want to go into the matrix. Um, but they do not because they can't get there. And the only way they can get through is by going through ATP synthase. And so here, ATP synthase utilizes the downward flow of those protons into the matrix and draws off the energy in order to generate ATP from ADP and inorganic phosphate. So this is using the potential energy of the proton gradient that was generated by the electrons. And so that is the chemoosmotic mechanism, the use of this proton gradient in order to make ATP from ADP and inorganic phosphate via the protein ATP synthase. And we call that potential energy the protomotive force, proton motive force. Um, and there we go. Look at that. We just broke down glucose. So we started with glycolysis. We took this molecule glucose that represented potential energy. We split it into 3 carbon pyruvates. 2 of them generated 2 molecules of ATP net here because we had to invest 2 in the glycolysis process. This occurred in the cytoplasm without oxygen. All right. We then took that pyruvate, pulled it into the mitochondria, and chopped off first carbon dioxide to form a 2 carbon acetyl-CoA. You generated some NADH in the process. That NADH goes to the electron transport chain. This acetyl-CoA feeds into the citric acid cycle, or the Krebs cycle. And there you generate 2 molecules of ATP. You generate 4 molecules of CO2. And then you generate 6 molecules of NADH and 2 molecules of FADH2. So, a little bit of ATP, lots of these reduced electron carriers. You take these reduced electron carriers, you feed the electrons into the electron transport chain in the presence of oxygen to form lots of ATP. I don't think I've said this before, but you can get 32 molecules of ATP from oxidative phosphorylation. So much more efficient than than substrate level phosphorylation. And we talked about sugar and glucose going into this system, but you can also get to it from other ways. So fats can feed in directly into acetyl-CoA production via the beta oxidation of fatty acids. The glycerol kind of goes into glycolysis. Proteins get chopped apart and they feed into various aspects. So there are all sorts of entrees into aspects of the Krebs cycle. Ultimately, they all go to that acetyl-CoA group. Um, And this process um, occurs. So here's kind of the bean counting here. I'll say this process stops without oxygen here at glycolysis. You get some ATP out, right? Just 2, not the 32 that you get before. And this pyruvate has to be processed. In animals, it gets processed to lactate where it leaves until it can get reformed into glucose. Um, and in yeast, you actually form carbon dioxide and leave a 2 carbon compound, ethanol. This is the carbon dioxide that's formed from yeast fermentation, is why bread rises, or you get carbonation in soda. It is the carbon dioxide formed from that fermentation process that allows that to happen. And then in presence of oxygen, you go through pyruvate oxidation, citric acid cycle, electron transport chain, and the total is 32 ATP out of this. And let me see. Oh, I didn't have a nice little um, metabolic map here. But then this is that whole process that we've talked about. And as I said before, you can, in various places, go out to form lipids. You can go out to form your nucleic acids. You can get some amino acids, all going from various places in the citric acid cycle. And then if you look at this, the full oxidation of glucose, so going from glucose to carbon dioxide and water, yields 280, 2000, 2,800 kilojoules per mole. Uh, Whereas from glycolysis, you only capture 146, right? So you leave a lot of energy on the table with this by not going through fermentation. And there's some organisms, I mean, by not going through oxidative phosphorylation. There's some organisms that can't, obligate anaerobes that can't undergo use oxygen in this process. And we won't talk a lot about this, but it's pretty neat what happens with pyruvate in the absence of oxygen. Here, we actually end up reducing that pyruvate. And so we use a lot of energy to go back to kind of where we went before to make this lactate. And this allows for the regeneration of NADH during, or NAD plus during glycolysis in the cytosol. Um, So I will stop. Well, no, I won't. Just kind of a couple more things to talk about for regulation. Obviously, this is a pathway that deals with energy dynamics. So the regulation dynamics uh, is going to be important, and it's going to be regulated by the energy needs of the cell. And so if you think about it, if you're sitting at rest, and your cell has lots of ATP, then it's going to send a signal saying, I don't need more ATP, right? So if it has lots of ATP, it's going to stop this cycle. Whereas if it has lots of ADP, so the cell is being used, it has lots of ADP, and it's going to send a signal saying, I need more energy. And so this kind of makes intuitive sense, right? So this pathway at various points will be inhibited by concentrations of ATP. So increasing concentrations of ATP blocks the pathway to make more ATP. On the flip side, it's going to be um, enhanced or activated by increasing concentrations of ADP because that says, hey, cells at use and I need more energy. And so that's going to drive the process. And so here you'll see in glycolysis, inhibition by ATP concentrations, enhancement by ADP concentrations, and that tends to work at the level of phosphofructose kinase because that's where the cell makes a decision as to whether to invest that second ATP or not. Stop that. And you see that again at the citric acid cycle. Inhibition by ATP or NADH, which shows that the cell is at rest. Activation by ADP or NAD plus. Okay, Oh yeah, here's some cool videos that are there if you want to look. I like this oxidator, I love it. I haven't watched that in a while. All right, and there.